Hi everyone, so today I'll be going over the JAK-STAT signaling pathway. This pathway regulates embryonic development and is involved in the control of a lot of processes such as stem cell maintenance, hematopoiesis, and your inflammatory response. So before we go over the pathway, I just wanted to go over a few things involved in the pathway. The first of which is cytokines. And cytokines are just the signaling protein for this pathway, and it acts as the ligand. And the receptors that are used are the cytokine receptors. And cytokine receptors can bind specific cytokines, but they can also bind hormones. And they usually are in the form of dimers or trimers and are associated with typically two JAKs. There are a lot of different cytokines that can be used in this signaling pathway, and you'll see a little bit later on that a lot of other pathways can converge with the JAK-STAT pathway. So a few of the cytokines that are used can be granulocyte macrophage colony stimulating factors. So that's involved with your immune system. And then there's two groups of cytokines that are pretty common, and they're separated as TINs and TERs. So the TINs can include erythropoietin, prolactin, thrombopoietin, and leptin, which we've heard about before. And the TERs that are cytokines are the interleukins and the interferons, and those are also used within the immune system. JAKs are kinases. JAKs is short for Janus kinase, and they are cytoplasmic tyrosine kinases. And here's a little picture. JAKs are associated with the receptor, and they have a few domains, which is a bit more in detail than we probably need, but just for your understanding, one of the domains, SH2, it binds to phosphotyrosine residues, and we'll see later on how phosphotyrosine residues are important in this pathway. And the SH2 domain also aids in activation and dimerization of STAT, which we will get to after this. The firm domain is responsible for receptor binding and protein-protein interactions with the membrane proteins. So as you can see, the JAKs are attached to the receptor. And then the PTK domain is responsible for the phosphorylation of tyrosine residues. So STATs are signal transducers and activators of transcription. They're also called latent transcription regulators because in their inactive state, when they're not phosphorylated, they're in the cytosol, and then once they become phosphorylated, they move into the nucleus and activate gene transcription. I should mention that different JAKs and STATs are recruited based on tissue specificity and receptors engaged in that signaling event. Okay, so now we can go through the pathway. So the pathway starts when a cytokine binds to its corresponding cytokine receptor. And the receptor, as previously mentioned, is already a dimer or a trimer, but the binding brings them a little bit closer together so that the JAKs are closer together, and they undergo self-phosphorylation, or they'll phosphorylate each other, and that's usually called transphosphorylation. And after they phosphorylate each other, both of those kinases are now active, and they will in turn phosphorylate the tyrosine residue on the cytoplasmic tail of the receptors. This phosphorylation event essentially makes a docking site for STAT to come in and bind to. And so when those tyrosine residues on the receptor are phosphorylated, STAT is recruited, and STAT's SH2 domain mediates the binding of STAT to that docking site. I'll show you guys an image a little bit later on, but essentially when JAK phosphorylates itself, that increases the activity of the tyrosine kinase domains, and that can lead to the activation of other signaling pathways, such as the AKT pathway or the RAS-REF-MEK pathway that we've heard of before. The pathway components are regulated by members of other signaling pathways. So that could be ERK or MAP kinase or PI3. In addition, when JAK phosphorylates the receptor and makes that binding site for STAT, there are other adapter proteins that will bind to that docking site and couple receptors to the RAS-MAP kinase pathway. So that's kind of how this pathway diverges or converges with other signaling pathways. But after STAT binds to that docking site on the receptor, JAKs will phosphorylate the STAT on their tyrosine residues, and that phosphorylation event dissociates STAT from the receptor, and it's back in the cytosol. And from there, it can bind to another phosphorylated STAT protein, and that will create a dimer, a STAT dimer, and that dimer will translocate to the nucleus and bind to specific sequences and stimulate the transcription of genes. So this was a really basic overview of the pathway. It's obviously not that simple. It's very complex, and it kind of intertwines with other pathways. This image might be kind of hard to see, but you can see the AKT pathway on the far right, and how JAKs basically stimulate the activation of other pathways. The pathway is regulated by negative feedback and also something else that we'll get to. So when the STAT dimer translocates into the nucleus and initiates transcription, somewhere along the line, regulator proteins are transcribed, and those help regulate the pathway. The regulator proteins determine the initiation, duration, and termination of the signaling cascades, and some of those include SOCs and PIAs. We'll get to PTPs, but the ones that are transcribed by STATs are the SOCs and the PIAs, which are suppressors of cytokine signaling and protein inhibitors of activated STATs. So both of these regulator proteins can only work when the pathway is activated, and that's important. So to go into that regulation a little more in depth, we'll start with SOCs. So as I just said, SOCs are transcribed as the result of STATs initiating transcription, and the role of SOCs is to bind to the phosphorylated tyrosines on the receptor. Those were phosphorylated by JAK, and those compete with STATs in binding to the docking site on the receptor. Also, it can bind to the phosphorylated tyrosines on JAK itself, and it can inhibit the kinase activity and thus the recruitment of STAT. PIAs, on the other hand, bind to the dimers of the activated STATs. So this is when the STAT has been phosphorylated by JAK and it dissociates from the receptor and binds to another STAT to make that dimer. PIAs bind to the dimers and inhibit them from binding to the DNA sequences in the nucleus and essentially inhibit transcription and act as transcriptional co-repressors. Negative feedback isn't enough to turn off the response, so the activated JAKs and STATs must be also inactivated by dephosphorylation of their phosphotyrosines, and that's done by the PTPs, which are the protein tyrosine phosphatases, and these will basically bind to JAK and STAT and remove their phosphate group and render them inactive, so signaling is halted. Here's an image of those regulator proteins. So at the top, you can see The SOC proteins are inhibiting the JAK kinase, and also the PTPs are inhibiting JAK by binding to it and removing its phosphate group. And it's actually SOCs that tag JAK for ubiquitination and send them to the proteasome to be degraded. But you can also see the PTPs binding to the STAT dimers and also inhibiting transcription in the nucleus. And you can also see that the PIAs are inhibiting the STAT dimer from binding to DNA in the nucleus. So this is a pretty good image in showing all the different pathways that are kind of converged or diverged from the JAK-STAT pathway, but it also includes a lot of the regulator proteins. So the green one towards the top by the receptor, the SHP1 protein, that is a PTP, so that is a phosphatase that's removing a phosphate group from JAK and making it inactive. You can see the PIAs in the nucleus inhibiting the STAT dimer, and you can also see the SOCs being transcribed, and then they go back out into the cytoplasm and get translated and then go inhibit JAK from there. And you can also see it competing for that binding site on the receptor. So this concludes my explanation for the signaling pathway. Let me know if you have any questions, and I'll be sure to share these slides with you guys.